super excited to be here. And as you have questions, I find it's easier to just plop them in the Q&A box right away, and then hopefully we can address them throughout the webinar today. I was tasked with giving you the disease basics and overview of bovine leukemia virus today. And then I'll be joined, Phil will talk about some of the consultations and ways we use the data that I will hopefully set up foundational knowledge for his presentation. Let's see. I have it on this slide, and depending on what your view looks like, there should be a black box with a smiley face. There will be no content or anything important in that black box. So if you need to move part of your video imagery or the control panel, if you put it up here in this upper right hand corner, you're not going to block anything on the slides. So I'll give you a minute to get adjusted, and I'll tell you what, I'm going to do the same. Hopefully I'm still a little rusty with Zoom. It's been a little bit since I used it. Okay. So today, my thought was to cover the 5 W's and the 1 H. The who gets BLV, what is it, where is it coming from, when, why, and how do we manage it? In any corner where you see the Spartan helmet from Michigan State University, that's going to indicate work that someone, whether it be my team or teams before me or at the same time, but it's work that's done through Michigan State University. And I did use a lot of QR codes, or I have some QR codes. So if you want to link to the references, you might have your cell phone handy or a way to get those QR codes. And of course, I'm happy to share them with anyone to distribute later. So let's just jump right into it. What is bovine leukemia virus? It's a Delta retrovirus. And what a retrovirus does is it integrates, when it infects an animal, it integrates itself into the host genomic DNA. So the virus itself is an RNA virus, goes into the cellular mechanisms of the cow or the animal that it's infecting, and it integrates itself into the cow's DNA or the cow's genome. It likes to target B cells. And when it does that, so B cells are a cell within the blood. So this is a blood-borne virus, or blood-to-blood transferred virus. When the viral DNA integrates into the host DNA, then it is called a provirus. So you'll hear me talk about proviral loads a little bit later in this presentation. That's what I'm talking about. It's the virus when it's integrated into the cow or the host genome. So it targets B cells. The virus targets B cells. B cells are responsible for the adaptive immune system of an animal, and that they produce antibodies to fight pathogens or infections. What happens when a B cell is infected with bovine leukemia virus, or BLV? The signals are not received. Those B cells are not as functional as a healthy B cell. So the signals are not received by B cells. There's improper maturation of the B cells that are infected with BLV. There's decreased antibody production and really a compromised immune memory and a compromised immune system, which MSU, Dr. Cousins had done some work a few years ago, and I'll show you some of those graphs from his research. Bovine leukemia virus is the virus, and you might hear bovine leukosis. Bovine leukosis is what the animal succumbs to, or is the disease, if you want to think of it that way. When do cows get it? There's vertical and horizontal. I'll just throw it all up here, vertical and horizontal transmission. So think anything that's blood-to-blood transfer. This virus is in the cells in the blood. So we can have placental transmission. The virus is small enough to go through the placental, so from dam to calf. There's blood-to-blood or blood exchange at birth. And then I have colostrum and milk on vertical and horizontal transmission, in that depending on your research area or who you talk to, they can be considered vertical or horizontal. Vertical if it's the dam and the calf that are the parental pair, but sometimes on farms we have mixed colostrum, or the colostrum in milk is not coming from the dam herself, it's coming from somewhere else on the farm. That then would be horizontal transmission. So the virus is in that colostrum or milk, it can pass then into the offspring. If it's frozen or pasteurized colostrum, it's processed, the virus is not alive, or it's not able to infect the calf at that point. Horizontal transmission is then between herd mates, and think anything blood to blood transfer. So biting flies, they've found BLV in biting flies. The use of needles and sleeves, natural breeding, dehorning and tattooing, and foot trims are all areas, more common areas that in research that we found there's blood to blood transfer. So there's bovine leukemia virus transmission. My slides won't advance. This is good. There we go. So who gets infected? This is a group of dairy producers or dairy interests. So it's infected in young stock and adult animals of dairy animals. Beef animals are not excluded. Neither are bison, buffalo, yaks, camels, and sheep and goats have also get infected with BLV. Sheep and I believe goats also, once they're infected with bovine leukemia virus, succumb to the virus fairly quickly, where cattle, it can render, it stays hidden from the immune system, and it doesn't lead, not every single animal succumbs to bovine leukemia virus, not every single animal gets the tumors, but in sheep, it's a different story. I just wanted to put that out there, but I know this group is dairy focused, so we'll stick with the dairy animals for today. So why should you care? And this is where I'll spend most of my time today, and where I bring the research and what our group or people, individuals of the MSU Extension and Research team have found. So there was a study that looked at 3,611 dairy cows from 91 different herds across 9 states. And I think Phil references a part of this study too in his slides coming up. They found positive cows, so cows that were infected with the virus, were 30% more likely to be culled during this 29-month monitoring period as compared to BLV negative animals. And so just to set up the future slides like I did in the beginning, the QR code down in the bottom right-hand corner, if you scan that, it'll take you to the reference. So this is a published article, peer-reviewed article from Dr. Bartlett did this work, and the Michigan State helmet, because it's Michigan State work. The other thing I mentioned was that animals with BLV infections, they have a compromised immune system. So this was a Dr. Cousins study, and what they did was they took BLV positive and negative animals and gave them a vaccine, and then looked at titers or antibody responses to those vaccines. The dark bar, these are just figures from that paper. Again, if you want to read the paper, look at the figures and the descriptions. That QR code's in the bottom right-hand corner. So the black bars on this graph are the percentage of cell types found in BLV negative animals of that study, and the light gray are the BLV positive animals. So CD4, CD8, or the cells represented in the red box here, you can see they're T cells. So T cells are involved with directly killing cells infected with a virus or recruiting other immune cells when encountering a pathogen. And you see that all three of those cell types are had a lower percentage in BLV positive animals. So they're not having this, they don't have the higher percentage or as many cells that are involved with killing other cells infected with virus, that compromised immune system. These bars here are of B cells, and we see that the BLV positive animals had a higher percentage of B cells in the study, but that's not a surprise. One, it's the first line of defense when encountering a pathogen, and these animals were selected. We knew they were BLV positive, which is a virus, so that the animal's already mounting an immune system and trying to fight off the virus. So we're not surprised that they have a higher percent of B cells. But when we look at the response of antibodies to the vaccine they used was the Bovishield Gold. So these are all incorporated into that vaccine. So these are what you're looking at on the lines is the antibody response, the OD, from the vaccine. So BLV negative animals are these. I don't know if you can see my mouse, but BLV negative animals are the solid black line, and BLV positive animals are that light gray dotted line. And across all three of the components that were in the Bovishield vaccine, you see a lower antibody response in BLV positive animals. So they're not able to mount, animals with BLV, infected with BLV, aren't able to mount that immune response and be protected from the vaccines like a BLV positive animal would. That's not all. So we also found Dr. Norby and Erskine Bartlett, this was a Michigan State study as well, found that the animals with increased levels of bovine leukemia virus have decreased milk production. The study out of Canada found that animals with BLV higher levels, lots of virus circulating in that animal system, they had lower milk components. And just to not make it all MSU study focus, our research is also supported by others, or others support what we're finding as well, that animals with BLV are likely to be culled earlier. They have lower milk production if they're infected in the first and second lactation. That's a study out of Canada. And then while it's a study in buffaloes, they found decreased reproductive performance and lower milk production in buffaloes in Egypt. To date, all the research that's looked at reproductive success in animals in BLV, I haven't seen anything with dairy cattle, but I wanted to be complete in what I've shared with the literature. And this isn't it. This is just a snapshot of some of the research that's out there. Recently, we had a master's student graduate, and I say recently, it's probably been 2 years now, that they did a partial profit loss with some of the farms we've worked with, and they estimated the BLV impacts on a per cow basis, which, because BLV affects the welfare of the animal and they're immune compromised, they're more likely to be sick with other illnesses. So sometimes it's hard to toss out, especially when you're looking at budget, piece out what's due to BLV, what's due to some other things, because it's integrated within that one animal. So we tried to in this study, and we looked at things such as vet labor costs, drug costs, breeding costs, and things like that were on the farm. And we estimated an average partial profit loss of a BLV positive cow to be somewhere between $200 and $400 per lactation. So hopefully I'm showing you evidence of why you should care. I have one last thing to bring up, and that's the condemnation. So when an animal is submitted for slaughter, 22 and a half, and this was an estimate or records from 2009. So I apologize, that's the most recent one I have. But 22% of the cow condemnations coming through the slaughter plant or the harvest location were condemned due to lymphoma. And of that, the dairy cow condemnations of dairy cows was 26 or almost 27% due to lymphoma. You know what and how can it be combated? So one thing, it's all right, I'll get there. I think my slides are missing some slides, but that's okay. If they're not in there, I'll go back and cover some bases. So bovine leukemia virus there's two ways to test for it or diagnose One is ELISA and the other is through qPCR or PCR. ELISA historically has been the test that's been used. It detects antibodies to BLV, so it's a seroconversion in an animal. And there's multiple samples that you could use and run ELISA on or submit for the lab to run ELISA, and that's milk, serum, or plasma are the most common. What an ELISA does is it detects the presence of antibodies or that seroconversion. So while it says the animal was infected at some point with BLV, it's not indicative of a current BLV infection or a new infection. It's low cost. The results come back fairly quickly, but it is unable to discriminate between maternal passive antibodies and active immune response. So if you talk to anyone that's running the ELISAs or the labs, they'll suggest that you wait for the animal to be old enough that they have an active immune system and they're not protected by those maternal antibodies, because the ELISA doesn't discriminate between the two. The qPCR, and I don't want to steal some of Phil's thunder that you'll hear from him later. I'll just set up what it does and what it's telling you. So PCR is they're actually amplifying that provirus. So the provirus again is bovine leukemia virus that has integrated into the host DNA into a B cell. It's DNA, it's viral DNA. That PCR detects the DNA of the virus. Typically, we use a blood sample to run that assay. You could use other biological samples, but since the virus lives in B cells, which is a blood sample is the best sample to use when running PCR. It's BLV specific. It's able to be performed on multiple sample types, like I said, so anything that you can extract DNA from. It has undetectable levels are possible. So you get a quantitative result. So essentially how much provirus is in that animal or how much virus is the animal harboring. The downside is you wait a little bit longer for results. It's a longer assay to run and it may be cost prohibitive. So we run samples, or we've collaborated with Central Star, and ELISAs are on the range of $4 to $6, and qPCR is more like $10 to $15. Here's an example. Like I said, we've worked with Central Star, and this is just an example of the output that they provide. This is what you would get as a producer or veterinarian after you submit samples. Your ELISA results come back as positive, negative, or suspect. So in that final results column all the way on the right, you see they're all N's or negatives. The test value is the optical density of the ELISA, and really, we haven't found anything related to or associated with proviral load at an OD value. So just wanted to let you know that's what it was, but it doesn't correspond to amounts of virus or anything like that in these ELISA tests or assays. Where qPCR gives you that proviral load, so that the sequence or amount of virus. You have an undetected, low, moderate, or high. So Central Star categorizes how much virus is in those animals into those categories, and that's what they give you as these final results. So down here in this bottom table in final results, you see one animal was undetected and one was low, so low amounts of virus. The test value is that proviral load. So the way this is a ratio of animal cow DNA to viral DNA. So the larger the number, the more virus you have in that animal. And you see 0 is undetected. They were unable to detect or have a threshold of virus in that animal. And then low is this 0.06. High would be above 1. So one thing that we've done when we talk about how this can be combated with bovine leukemia virus, and Phil will go into a little bit more detail on these, so I don't want to steal his thunder, but I'll set up a little bit about what our team has done. We lead consultation discussions and do herd scans. So part of the consultation discussions include the BLV cow list, so you know or can rank animals on how much virus they have. We look at the herd BLV prevalence to have those discussions. Tracking over time, you identify new infections and maybe where those infections are coming from. We look at prevalence by lactation and shedding by lactation. Shedding, when I say shedding, I'm referring to the amount of virus in an animal that's infecting herd mates or potentially infecting their herd mates. Distribution of shedders. So again, shedders are those animals with higher levels of virus that are more likely to infect their herd mates versus animals with lower amounts of virus. And then we go through management notes. So what are things that we could do or that the farm could do, the veterinarian can do that will combat some of the bovine leukemia virus infections that are happening on the farm? Things we talk about. Again, I put Phil's picture here to remind me that Phil's gonna hit on some of these things, but things to consider or things that we talk about are day-to-day management things. So the testing, what are you doing with needles and sleeves? And if you're unable to use a single-use needle, maybe there's other management strategies that we can discuss that would help, or not do the blood transfer from positive to negative animals to cause more infections. Maybe it's a third strike. So we know that when you're selecting or when farmers are selecting for individuals to keep on the farm or retain and get rid of, there's a host of traits that they're looking at. Maybe BLV infection could be one of those strikes against the animal, and proper hygiene on the farm. We also talk about big picture management, so testing and culling animals, monitoring animals, segregating them, placing positive ones on a do not breed list or breeding to beef, reducing the number of purchased cows, and then testing those purchased cows. And I'm sure in discussion, and if you have other ideas or questions, I'm sure there's other management strategies that that could help. These are the ones that we go to when we start those conversations with producers. One thing, and this is I like to say hot off the press. This is Madison, my PhD student, and she's been working on the last 5 years. We've been following calves on the farm and sampling them to see where in the life of a young stock animal infections are coming from. And I'm here to tell you that every farm is a different story. BLV solution and management is not one solution fits all. And that's why these consultation meetings and conversations are so important. So we followed 5 farms for the last 5 years, and each farm has a different story about where those infections might be coming from, or what's the best management strategy for that farm. One thing that we did find with her data set, though, was that calves born to a BLV positive dam were 3 times more likely to be BLV infected within their first year of life. And this project all started from the consultation meetings we had with farmers. They noticed that, and they noticed that they had positive dams. The calf was more likely to be positive. So we just put the research behind it. We also found that daughters of dams from a higher proviral load, so animals that had higher viral levels, their daughters had a greater likelihood of testing positive for a higher proviral load category in their life as well. So dams that had high proviral load, their daughters were more likely to have high proviral load in their life as well. So daughters born from ELISA positive or high proviral load dams have an increased risk of being affected by the virus as well. You might ask why. Granted, that will be the next study, but we do know there is literature out there, and some that our group has done, that there are genetic markers for resistance and susceptibility to BLV. Whether we didn't sequence these animals, so I can't comment on that with this particular study, but there are other studies. We know that there's genetic markers that are resistant to bovine leukemia virus. So while the animal can become infected, they never get up into the higher categories of PVL. They never reach the high levels. The lower levels stay maintained in that animal through its lifetime. The one thing, and I'm kicking myself because I don't know where my slides are. Before I close, there were a series of slides about BLV progression and how the disease progresses. So when an animal becomes infected with BLV, let's say a few B cells become infected, and that virus may lay latent, meaning that those cells don't divide. The virus stays in low levels, and that animal can stay in low levels of that provirus, that virus laying latent for its entire life, and would never have any issue. There are animals that once they become infected, the B cells get infected, those B cells start to divide, the virus also infects other B cells, and all of a sudden that animal escalates to a high proviral load category. And when that animal has a high proviral load, if they survive, they maintain or keep that high proviral load. We don't see a lot of animals that bounce back and forth from high viral loads to low and then to high again, or medium and moderate. We see animals that they maintain whatever that level is until they don't. And there's some switch, and we still don't know what that switch is. There's some switch that triggers it, and the animal maybe has low amounts of virus for up to the third lactation, and then the fourth lactation something and the switch happens, which, welcome to research, we need to figure that out. But the switch happens and that animal could succumb to the virus, become high proviral load, typically growing tumors at that point and getting lymphosarcoma. And I apologize, I had slides with diagrams of that, and I don't know where it went in my slide deck. But it's one thing to think about of how that virus progresses. And animals with low proviral loads just may stay low until one day they don't, and then at that point it's too late to save the animal. That's really all I had. I'm open to discussion. Hear what questions you have, or something that was unclear that I could clear up for you.
Are there any questions from anyone out there? We don't have any questions in the Q&A yet at the moment, but I had a couple of questions. So you just touched on in that slide previous to this one about essentially genetically susceptible or genetically resistant cows to this disease. Are there any other kind of genetic markers that have come about, any kind of like health traits that may increase or improve a cow's likelihood of surviving this or fending it off?
That's a great question. So the genetics work to date has really just focused on a particular region and a particular gene, and that gene is the one that how do I want to? It's BOLA or MHC region, and so it's responsible for the antibodies and creating an immune response. It's one of the health genes that they use in the Holstein prediction and the indexes. Research to date has not looked for, to answer your question, no, there's no research that has looked for other genes or other health traits that are associated with this that might help. But it's a great question, and if we ever get research dollars back, it's something that would make a really nice project. All the projects have really just focused on one gene, one region in that BOLA gene.
Okay, I have another question about what kind of, I don't want to take away Phil's thunder if he's got this in his presentation, but
we'll give him a challenge. It'll be okay.
What kind of prevalence do you see in herds in general, and does prevalence vary depending on herd size?
Yes. So let me try to go, if I have a little bit of time, because my slides wouldn't advance earlier. I wonder if I did. There they are. So here, if I can take a few minutes. So this is the disease progression diagram. I did have this slide. So you see, we have a few of the cells that become infected. The animal becomes leukemic, low, small amount of virus. 30 to 40% of those animals then move into persistent lymphocytosis. So there's more cells that are infected. And then very few go to tumors, and at that point that animal's unwell or deceased. Where is it and on the herd? This kind of gets to your question, and if I don't completely answer it, make sure to ask again. But 94% of the dairy herds have at least one animal infected with BLV. The average herd prevalence is about 46%. We like to say 50%. It depends from farm to farm. I've seen very large farms that have a low prevalence. I've seen small farms that have a high prevalence. And Phil, it's okay if you wanted to unmute and comment on some of the things that you've seen as well.
Yes, indeed, I've seen high prevalence in herds. I've seen up to 90% prevalence in a herd, and it doesn't seem that farm size is the issue. However, I'll mention some negative herds, and it may be that negative herds typically tend to be smaller because they haven't purchased animals. So because it can be purchased in, if the few percent that are negative typically tend to be smaller, but once a herd is infected, then size is not an issue.
Okay. Do you have any other slides you'd like to still share?
No, I'm so sorry. They wouldn't
okay.
advance, and then I guess welcome to computer and technology problems. Huh? Let me get to the end and then. Oh, goodness. Yes, I do have two more slides I would like to share. So we have, and apparently they're on automatic. Oh my goodness. So we have a virtual conference this year focused on BLV, July 16th, and I put it in your time zone, so it's from 10 to noon. We'll have flash research talks, so very quick snippets of what the research team is working on. And this is not just us, this is international. So you'll hear the folks from Canada, Argentina, Japan. They're all working on BLV, and we all come together once a year. So we'll have some flash talks from 10 to noon on July 16th. On the 17th, producer-focused discussion. So if you'd like to come to the table and ask questions, share concerns, listen to others, the ones that we consult with, hopefully will join, have an open forum for discussion for the producers, veterinarians, anyone that's on the ground working with BLV and the animals or wants to learn more. And then the 18th is focused on building research collaborations and writing those next projects. So QR code is for registration and more information in the bottom left-hand side of your screen. And if you're would like to join the BLV listserv, there's an email listserv. You can scan this QR code. And if you don't want anything to do with QR codes, feel free to send me an email. My email is down here in the bottom right-hand corner. And I'm happy to share any of the resources, connect you, or answer any questions that come up that maybe don't come up today, but tomorrow when you're stewing about BLV.